Today we are going to do isolation of DNA from onion cells. For it we would need filter paper which is stapled using a stapler. Then we would need a dishwasher which will help in separating the cell membrane, which is a lipid bilayer. Then we would need sodium chloride. Then we would need, then I have used a stirrer as an aluminum rod. We can use glass rod instead. And I have mixed the salt and this dishwasher into a solution of water. Then we will use onions, which are the main ingredients. We should use ethyl alcohol, but ethyl alcohol is not very handy to get, so we will use propanol, which is ethyl alcohol must be 90 percentage above, but we didn't get it, so we will use 80 percentage propanol, which will help in separating the DNA, isolation of DNA from the onion cells. Now we are going to grind this in the mixer grinder. Okay. Now we have grind the onion really well, and we should add some tablespoon of onion into the glass. Then we must stir it really well. Okay, we can add some more. It didn't make up the quantity. As we know that this glass contains the solution of detergent, dishwasher, and then sodium chloride. Now I have stirred the solution really well, and I think it really combines really well, and I am going to heat it at 40 degrees Celsius, but I do not have any measuring device to measure the temperature, so I am going to use preheated water. Going to filter this solution which has heated over the preheated water. To the chilled, now I am going to add chilled ethanol to the solution. Now stir it really well. Sorry, I have made a mistake that I have added propanol into the solution, but we can see that it worked really well, that we can see the DNA is precipitating out of the solution, and we have to stir it really slowly so that we can see more and more DNA is getting precipitated out of the solution. And this is the DNA is precipitated out of the onion cells. So we can see the difference when I placed it in front of the sunlight. We can get the DNA, you can really visualize through it that a cloud like structure is present and it is getting precipitated more and more. This is DNA. So dear friends, as I got the DNA, as I didn't expect this much to be effective by using propanol. So you can try it out at home using these simple ingredients, and also homemade. You can just precipitate out the DNA really easy without using much lab equipments. Uh, So I got the DNA from the onion cells separated really well.